This video, and it's going to be a short one, is going to introduce you to molecular biology. In this first video, because this is going to be a short series, we're going to be reviewing what you already know about DNA and RNA, and we're going to talk a bit about DNA replication. We'll continue in other videos. I rather don't have my videos too long, if I can help it. So, what is the central dogma of molecular biology? Central dogma says DNA encodes for RNA. Remember, DNA is a molecule that contains all the genetic information. Passes the genetic information to RNA, which in turn passes the information to proteins. Proteins are the ones who do everything in your body. Okay, so the DNA stays in the nucleus, very well protected, because if you damage the DNA, that organism will ruin, and sends information via the RNA to make proteins outside of the nucleus. There's two different processes. Transcription means passing information from the DNA to a messenger RNA, and that takes place in the nucleus. I like to compare DNA to a cookbook, and you have lots of recipes. But if someone asks you for the recipe for lasagna, you don't copy the whole cookbook, do you? You just copy the recipe for lasagna. So that's pretty much what the DNA does. DNA has the recipes, the information for every protein that the cell may want to make. But the cell wants to make one protein. For instance, a red blood cell, and it needs to make hemoglobin. There's no need to send information for everything out. The DNA makes a copy of the recipe for hemoglobin, and the RNA comes out of the nucleus, goes into the cytoplasm, and makes hemoglobin. So, transcription goes from DNA to RNA. Translation is the process of getting information from the RNA and making the protein. And that takes place in the cytoplasm. How do you remember which one is which? It's very easy if you think of an example. Imagine a courtroom. You know there's always someone typing everything that's been said. So the judge says this, and the lawyer says this, and the person says that, and all of that. Hopefully the proceedings take place in English, and the person that's typing is typing it into English too. The final result is a transcription, a transcript of what took place in the courtroom. DNA and RNA are made of nucleotides. It's the same language. Now imagine the person who's on trial doesn't speak English. So you have a translator. And the translator hears the lawyer asking a question and interprets it into that person's language, takes the answer and puts it back into English. It translates from English to whatever language back to English. So translation means that you change from one language to another. RNA is made up of nucleotides, protein is made up of amino acids. So those are two different languages. Okay? That's an easy way to remember, I think. So let's review what we know about DNA. We talk about being a double-stranded molecule, double helix. It's in the nucleus. The sugar is deoxyribose. That's different from RNA. There's a sugar phosphate backbone. The blue ribbon in there is sugar and phosphate. You have 4 bases. Now, we're going to get into a little more of detail. We are grouping them into 2 groups, purines and pyrimidines. Adenine and guanine are purines. Cytosine and thymine are pyrimidines. You do need to know that. I'm not going to ask you to memorize the formulas. Okay? And remember, one group has 1 ring, that one has 2 rings. Because of the DNA base pairing rules, adenine always pairs with thymine, guanine pairs with cytosine. You will have a 1 ring pairing with 2 rings, so the thickness of the DNA molecule remains constant. And we are going to see very soon how important that is. Okay? Really, other than purines and pyrimidines, it's just a reminder of what you already know. Where do you find DNA in the cell? Well, it depends. In a eukaryote, you say you have a nucleus with a nuclear envelope. So the DNA is there because it's protected. Okay, so you have a drawing here. You have the nucleus and the chromatin, that's the DNA and proteins, inside the nucleus. In a prokaryote, you don't have a nucleus, not a nucleus surrounded by a membrane. You have a nucleoid, an area where the DNA is located. Okay. Now we talk about DNA must be tightly packed. The nucleus is very, very small. We talk about the length of the DNA in a molecule, so it has to be tightly packed. Once again, this is reviewing. The DNA wraps around histones, those are proteins, and the histones wrap around by DNA from a nucleosome. When you have a nucleosome and a little strand of DNA and a nucleosome and a strand of DNA, we call that beads on a string, because when you look under the electron microscope, that's what it looks like. Nucleosomes coil, you have a chromatin fiber that's actually DNA and protein, but if you continue coiling and getting thicker and more compact, you eventually have a chromosome. Basically, even when we talk about the beads on a string, you need to use an electron microscope to see it, because it's not thick enough, but a chromosome can be seen with a regular light microscope. You have condensed it enough, can be seen with a light microscope. Okay, but these are terms we're going to be using periodically. We say that the DNA strands are held by halogen bonds between complementary bases. Remember, we say adenine always pairs with thymine, guanine with cytosine. That's what they're called complementary. Okay, the two strands are also called complementary because the sequence of bases in one strand can be used to make the other strand. If you know that one strand is A, G, G, C, A, T, T, A, you say, okay, I can see what the other strand is going to be because I put complementary. T, C, C, G, T, A, A. I don't know what I wrote in there. Let's call it an A, T. You put always a complementary base. One thing that most people don't realize is the direction of the strands. Basically, when you look at the ribose, you have the sugar, phosphate, sugar, phosphate, sugar, phosphate. You have an OH in what's called the 3' location in there. Then the 5' is the one that forms the bond. And here the 3' is part of the bond, but you have an OH in the 5'. So that's how you come up with the 5' and 3' ends. And most enzymes are going to read the DNA always going from 5' to 3'. Obviously, the complementary strand moves in the opposite direction. Okay, and that's going to become very, very important. But with very few exceptions, everything that happens with the DNA is always going to be in the 5' to 3' direction. We did talk about replication. We say it was semi-conservative because out of the new DNA, one strand is going to be a parental strand, another strand is the newly synthesized. We talk about Helicase separating the DNA strands, forming replication fork, and now DNA polymerase gets in here. Let's look at the upper strand for a moment, okay? So you have the DNA polymerase in here. So DNA polymerase is going to make DNA in the 5' to 3' direction, okay? Well, first, it has to create an RNA primer. DNA polymerase cannot begin from scratch. So we're not going to worry about what enzyme, but it makes a little RNA, and now DNA polymerase is going to be polymerizing 5' to 3'. So yes, this is 5'. It makes a continuous strand. That's why that original strand here is called the leading strand, because the DNA synthesizes continuously. On the other hand, if you go in here, if you move in the same direction from right to left, you'll be going from 3' to 5'. And we said no. DNA polymerase has to go 5' to 3'. So what does DNA polymerase do? It jumps. And it makes a piece of DNA, because now it's going 5' to 3'. Yes, it does put a primer and all of that. And then it jumps again and makes another little bit. So working that way is synthesizing in the correct direction, but it has to keep jumping. It makes different fragments that are known as Okazaki fragments. The strand of DNA that when you copy, you get Okazaki fragments is called the lagging strand. And you say, yeah, but what could do all these fragments do? They're going to be joined together. There's still one more enzyme that's not shown in here called DNA ligase that will join these fragments, making one continuous strand. At the end of every chromosome, there's a number of nucleotides. Usually, these are repeating nucleotides, but it's called a telomere. Telomeres protect the chromosome from being destroyed by enzymes called DNAase. That's an enzyme that breaks some DNA, and it's present everywhere. Okay? So DNAase is going to try to break DNA. The telomere is like a fuse. Yes, it's going to allow the DNAase to break it, but it breaks it very, very slowly. While the DNAase is busy breaking down the telomere, it does not hurt the actual DNA. Now, usually the DNA is not exposed to DNAase, but when the cell reproduces, it happens. And during cell reproduction, cell division, part of the telomere is lost. But as long as there is telomere, it continues. And every time the cell divides, there's an enzyme called telomerase that adds to the telomeres. So yes, you lose a certain fragment of the telomere, but then you replenish it. And that cell can keep on living. If something goes wrong and you lost telomeres without replacing them, that's associated with cell aging, eventually cell death. Some researchers say, oh, if we could stop telomerase, then you're not age. Oh, would you still be 80 years old, but your body would be like the body of a 20-year-old. Wouldn't that be nice? So they did inhibit telomerase, but not removing the telomeres has been linked to cancer. No, it doesn't work. There may be a magic potion that will keep you young forever. We haven't found it yet. Okay? We talk about DNA polymerase repairing DNA. Basically, what it does is prove reads after replication. Sometimes it's mismatch repair. Look at that example. Accidentally, DNA polymerase put the wrong base. So you have a C in here, but in front it put a T. Anybody can make a mistake, right? Think about how long the DNA is. You make one little mistake. But DNA polymerase goes over, and all of a sudden it finds a little bump in there. Why? Because C and T, both of them have two rings, so all of a sudden the DNA molecule becomes a little bit wider at that spot. DNA polymerase says, aha, I made a mistake there. Well, the next thing is, which one is the mistake? One of the strands is the parental strand, so you know that one is right. So you go to the strand that's been formed and you say, okay, here's the mistake. DNA polymerase removes the wrong base and puts the correct base. Fixed. That's why DNA polymerase is said to make one mistake for every 2 billion bases it replicates. It might make more mistakes, but it fixes them. It's very, very rare for DNA polymerase to miss a mistake. Another common repair is when you get a Thymine dimer. Okay? Sometimes UV light is very good at that, causes thymine to form a bond with a thymine that's next to it. It needs energy from UV light. So what do? Nucleotide excision. Okay? Basically, you cut down that fragment and replace it with the correct fragment. So it's very easy to repair DNA in there. If you don't repair, what you end up is a mutation, a permanent change in the DNA. And we're going to talk about mutations at that time. Some are harmful, some are, well, so-so. There are beneficial mutations. We're going to find out that. Remember when we talk about the polar bears that have a mutation in one of the genes for fat metabolism? It allows them to eat fat without getting high cholesterol. So some mutations could be beneficial, but in general, the cell tries to avoid mutations, avoid changes. It's like, if it ain't broke, don't fix it. We talk about the similarities and difference between DNA and RNA, the two nucleic acids. We say DNA is double-stranded, RNA is single-stranded. The sugar, deoxyribose, the D for the DNA, or ribose, the R in RNA. And the basis would be similar, except that thymine is replaced with uracil. It's very similar, but it's not thymine. And again, I don't want to memorize the formulas. But in RNA, uracil pairs with RNA. Okay. Here you have the ribose and deoxyribose. Again, deoxy means lacking oxygen, so you can see when an OH is replaced with H in there. This is what we talk about the 3' and 5' end. You can polymerize here or there, but obviously at the end, there's going to be an OH either in the 3' position or the one on the 5' position. That's what we was talking about, the 3' and 5' ends of the DNA. There's 3 types of RNA, although we're going to see that there's actually many of this kind in there, but you have a messenger RNA. The messenger RNA copies the information in the DNA. I hate to use genetic coding here. That's not. Copies of a gene that'll be there, and it's going to carry that copy from the nucleus to a ribosome. Again, you want my recipe for lasagna? I'll write it down. I'm not going to write down my whole cookbook. I'll just write down that recipe and give it to you. Then you can go to your kitchen and make lasagna. But the little piece of paper where I wrote down the recipe, that's the messenger RNA. Now, if you're going to make proteins or you're going to cook lasagna, you need the basic ingredients. In the case of the proteins, you need amino acids. So you have transfer RNAs that carry the amino acids to the ribosomes. And we're going to see in another video that there's a different tRNA for every amino acid. Okay? And they're very easy to recognize because they have that clover shape. Finally, there's ribosomal or rRNA that combined with proteins is going to form the ribosome. So hopefully this is not difficult to understand, but again, you want to work one-on-one, you want to study for an exam, there's a link to my Washington account in the video description. Make an appointment and we'll get together and go over whatever you have questions about.